Hi, and welcome to our GCP-Mindset channel and all things on clinical research. Today, we have part 3 of our ongoing webinar series on risk-based monitoring for you. In part 1 and 2, we discussed the foundations of risk-based monitoring and what you need to know about its components. And today, we want to talk about its implementation. So, stay and listen. So now that we have sort of discussed the different components of risk-based monitoring, how do we implement it? Actually, of course, you need to start by having processes in place, and you need to have the necessary tools in place, and you need to have the necessary reporting paths in place to establish it. That's of course clear, and of course the staff needs to be trained on the new processes. These two steps were actually quite a challenge for a lot of companies as COVID-19 hit, because as I mentioned before, although there was a push for risk-based monitoring for the past 10 years and probably even longer, there was still not as much implementation of these methods. A lot of people still defaulted back to 100% source data verification. And not having the processes, not having the tools in place left a lot of people without options other than saying we hold the trials until we can visit the study sites again, or until we can develop the tools. For us, fortunately, that was not the case. And we really felt it was quite beneficial to us because we needed to argue less with the clients on whether to implement risk-based monitoring or not. There was a certain uptick in accepting that this is the way to go, because there was also a lot more regulatory reinforcement that this is the way to go. Nevertheless, if you have processes, if you have trained staff, it means that you can start your study by first assessing the study risks. And for that, you should go through all of the assessments, and I will also have an example in 1 or 2 slides. You go through all of the steps on where you collect data, where you process information from the patient, and where your engagement with the patient basically has an impact on the patient rights, on the patient safety, and on data integrity. These 3 core principles are always paramount for your clinical trials. And they should always be at the center of your risk-based strategy. And in case there is a question of, is it either impacting data integrity or patient safety or rights? Always the patient safety and patient rights are paramount. Once this is there, you then need to adapt basically a mechanism by which you can evaluate the risks within a trial. You need data sources for it. You cannot just establish risks, although recent studies also regarding the last 2 years have sort of investigated how much risk-based monitoring is picked up by different companies. And in the vast majority of the trials, the people started implementing study risks and identifying study risks, but that's where they stopped. The monitoring strategy was still 100% source data verification. And of course, you need to go a step further. First, identify risks, identify sources which can guide you through these risks, and then rationalize how you would like to do monitoring on it. More intensity, more frequency for high-risk processes, for high-risk data, less intensity and less frequency for low-risk processes and low-risk data. Now, if you do remote source data verification, you absolutely must also inform and train study sites. Some sites may be used to it, some sites just might not be used to it. And I can tell you from the DIA panel, which I was part of, there was a site management organization from Canada there, much more adept at also including information from through or having information verified through remote source data verification. And even they said that it is a struggle. It costs more time. Tools need to be implemented. The regulations at clinics, at hospitals, they need to be upheld. There needs to be IT support for this process. There needs to be training also on the other side, and that cannot be underestimated. If you're only doing remote or central data review, of course, it doesn't involve the study site, but it's still good to pass on information to the study sites and to let them know how you will monitor the data and how this process will be performed. And then the next step, of course, is monitoring according to your strategy, and continuously monitoring your risks and seeing if you need to update it. Here, overviews, globally accumulated, but also on country level and on site level, help you keep an oversight of how the status at the different parts is. Do you have a lot of missing data? Do you have risk factors that you need for your evaluation that are not included? Do you have randomization discrepancies? Do you have issues with the informed consent process? Is this high or low or medium at your study sites? How should you adjust the way you monitor your data based on what you see? Which sites, which countries are performing well? This is paramount to implementing and maintaining a risk-based monitoring strategy in your trials. Of course, this relies on the data sources you have. Sometimes you don't have data sources, then you need to make do with other things, with remote or rather with manual processes. And then it is difficult to assess whether you're in line with your expectations or whether something's at higher risk than you anticipated or at lower risk than you anticipated. So what we like to do is, in a first step, or maybe in a second step, but for sure during the planning phase, take what is usually part of a monitoring of a clinical trial protocol, and that is the visit schedule. Because the visit schedule gives you an extremely good overview over which assessments are performed at which time points, and is a very good basis for communication because it is very clear and to the point what is measured when and how. And you can utilize it to highlight for all participants in this collaborative effort, that is risk-based monitoring, which of the elements are really highly risky, which carry a lower risk, and which carry almost no risk. And of course, for the purposes of the demonstration here, I included 3 colors to make the shades slightly more distinguishable. Generally, if you assign risks to certain parameters, you could say that they're on a scale of 0 to 100 or 0 to 50 or 25, maybe at minimum probably 0 to 10, with different risk factors or risk elements included in there, which will be part of another webinar series we will be giving in around a month and a half or 2. But for sure, it's more diversified than just high risk, intermediate risk, and low risk. And how do you get to these risks? This is a very collaborative team effort. So for us, we combine people of various different departments, from the project management team to the remote data monitors to the monitoring team. The statisticians are involved, our product safety department and physicians are basically involved. And of course, also data management team can be involved here. So all of these people can basically provide input on how high the risk is of something going wrong. And this contains, of course, a few parameters. What's the impact if something goes wrong? What's the likelihood of something going wrong? And how easy is it for us to detect it? And if we have electronic sources for information, already the risk is a little bit lower. If it's a process that has no big impact, even if something goes wrong, the risk decreases further. And this really informs on the different elements of the trial. And now you can already see that even within a single assessment, in this example, I haven't given the same risk to all study visits. For example, if you're looking at the primary endpoint, and for example, you want to check the change from baseline and primary endpoint, in this case, I've just selected vital signs here, then the baseline assessment and the end of treatment assessments are much more important for the clinical trial than the other visits. And even for concomitant medication, where I would generally say there is no risk associated with it, I would say if you have very specific exclusion criteria that mean basically patients are ineligible if they take certain medication, then you should check much more closely before the inclusion of the patient. Everything that comes after is of lower importance. And this is how you can go through these as well. And it allows you to have a very differentiated view on the different types of information you include. Pivotal to implementing this is also understanding that the study phase will usually have a quite severe impact on your risk rating. Of course, don't quote me on the exact ratios now between the different types of risks, but in a phase 1, first in human trial, you have no prior information on patient safety. You usually, at least, you have toxicity and safety assessments performed in animal models, and you try to somehow apply the information gained there to your expectations towards the application in humans, but you don't know. And so during a first in human study, so phase 1 study, it inevitably also means you need to collect a lot of safety information. And that in all of the safety information, there could be a hint towards potential toxicities that you are just not aware of. And therefore, here, a lot more emphasis is put on a lot of the information that you gather. And as you progress through your trials, safety becomes less important. Of course, efficacy or performance or effectiveness for medical devices becomes more important. And eventually, as your drug is already released to the market and your patients can be treated with your medication anyway, whether they are in the clinical trial or not, then it becomes even less of a high risk factor because you have a very good idea already of what to expect in terms of the safety profile. And that is the basis of rationalizing why monitoring activities can be, let's say, limited, and why you can move away from source data verification and source data review and into remote monitoring. And then only for particular sites or only for particular subjects, you can do, again, source data verification. So following this first step of assessing the risks at different visits and for different assessments, then the next step is to assign and implement this risk-based monitoring approach. And this is what you see here. Again, visualizing it is a way of communicating it, which is why I particularly like this, because it gives a clear indication to the study team members exactly how and in which capacity data will be monitored. And you see here now that from no monitoring to source data verification, there's a lot of information included. And the trickiest part for communicating it, not with regulators, but with sponsors, is always to do no monitoring. Telling them that some data points, they can exist, but there's no importance to them, and therefore they should not be monitored. Not even by a remote monitor, not even by a program, because sometimes that is even not possible in the same extent. That is usually the most critical point of communication, but it's definitely worth it because usually in this information, and I will always use concomitant medication as an example, there's a lot of information contained which you may just not need at all. A medical monitor may look at it in specific cases to assess correlation, for example, to certain adverse events. But other than that, really cross-checking everything from the frequency to the way it was given to all of the other elements from start to stop date of the medication is a lot of work for monitoring and a lot of work that is spent that will not yield much information and a huge impact on data quality overall. One more thing that you can see in here is that sometimes we would also suggest to combine different types of monitorings. So you can see, for example, here where's my mouse? There's my mouse. For adverse events, and you can also see it here. In this case, this is, again, the primary endpoint in this example, here and here. We have both statistical monitoring as well as source data verification or source data review selected for these specific data points. And now you can argue that it doesn't make sense or doesn't this duplicate work. I would beg to differ a little bit. Of course, you do some amount of work twice, but statistical monitoring can have vastly different implications for you. For example, I mentioned before the example of site comparison of adverse events. That's something you would assess with tools, and that's something you should do in addition to source data verification, because this is an important data point for your study. So it can add information that you would not have by purely performing source data verification. And it is, of course, also something that will help you assess site performance. which again is a key factor that influences your risk-based monitoring approach. For the primary endpoint, I would always like to have the opportunity for a tool to also highlight certain values for me in case they were overseen and missed as someone performed associated verification at a study site. It can also inform me about trends that I may not be able to see maybe across visits or across study sites that I may not be able to see through source data verification. So finding a good rationale and using also the monitoring visit plan, the study visit plan, to really communicate your approach helps all of the stakeholders, because you will have different people performing remote monitoring, you will have different people performing statistical monitoring, and again, different people performing source data verification. So this needs collaboration, this needs communication, and having one good tool to communicate all of this is, of course, pivotal. And as we saw before, this approach, of course, scales. In this case, not with study phase, although indirectly so, but with study risk. The higher the risk, the more SDV-driven you may want your monitoring to be performed. The more risk, the less you want to rely on statistical tools. And sometimes you can even not do that. For example, in first-in-human trials, if you only have 10, 12, 15, or 20 patients, it is very difficult to draw information from statistical means because you need more patients, you need more study sites. If this is restricted, you need to rely on other elements. If you have thousands of patients, of course, the vast majority of information can be covered by statistical means, and then only a fraction can be done through source data verification if the risks allow for it. And as I mentioned before, if you have really low risk in your clinical trial, be brave enough to perform no source data verification. Yeah. In post-market clinical trials with medical devices, where there is absolutely no risk, or in vitro diagnostic clinical trials, you shouldn't uphold the same amount of stringency. These trials can be monitored just much differently than your pre-market clinical trials. Depending, of course, on the risk that's imputed, but especially with IVDs, they don't even come into contact with you. No safety risk. The only risk you would have is to patient rights to some extent. And there you can find good arguments as to why you may want to skip entirely on source data verification. So wrapping this up, where are we now? First of all, I hope I could show you that there is really a scientific and regulatory foundation that supports the use of offsite monitoring techniques, and that they are more efficient in that they basically provide you with the same quality, and that they still manage to uphold the regulatory requirements. Now, in addition to that, I think what the past 2 years have also shown us is that implementing a risk-based approach to monitoring is also preparing you in case there are again shutdowns, hopefully not through the next pandemic or continued worsening of the current one, but through unavailability of a study site, through time restrictions, because you don't always, after a patient enters or has a visit, you do not always immediately have a monitoring visit. And of course, therefore, it's also really good as part of your risk mitigation strategy. And this is also more and more demanded by regulators, to have a good risk mitigation strategy, to present evidence that you are considering limitations and that they will not impact data integrity and patient safety and patient rights if something were to happen. Current practice, well, I can tell you from our experience that the frequency with which risk-based monitoring and especially also offsite monitoring is adapted in clinical trials has, of course, drastically increased over the past 2 years due to the COVID-19 pandemic. I have last week been in a working group of the German Association of Pharmaceutical Medicine. And again, the same or a similar topic was discussed there, quality-based risk management, also with a small piece of risk-based monitoring. And one of the things that was of high interest, and there was a lot of uncertainty still across all of the 100 participants in there, was the question whether in implementation of a risk-based quality management approach, which basically is at the foundation of risk-based monitoring, is even viable from a regulatory point of view, from whether there's any expectations or experience with regards to inspections. And there is not a lot in there. Not a lot of people have been, for a study, inspected by the state, and the state has confirmed, yes, we do agree that your approach to risk-based quality management is in line with our expectations. It manages it well. I don't think you will also ever find really 100% of the confidence, because every study is unique, and you always need to find a way of justifying your approach. What we can say from experience, and we've done this a lot already in the past, and are still, of course, always pushing it these days, is that you can just implement it. You need to be prepared to have a very good rationale, and then you should also just stick up to it, because also not the inspectors have a lot of experience with this type of approach and exactly what they expect, because the regulations are so vague. So you will have individuals coming to individual conclusions on whether your approach is appropriate to their felt risk of the clinical trial or not. And that is something that you just need to be prepared to argue for. But definitely what we see is a higher rate of implementation of these practices in a day-to-day work, which also impacts everyone that's working in the area, from monitors to data managers to statisticians. There is also a shift, of course, that we can feel. There are perceived limitations, as I said, some remaining insecurities around regulatory compliance of this approach. I'm specifically saying it's felt or perceived limitations. There is, and I'm pretty sure there will never be, exact specific regulatory guidance as to how to perform this process. I think the biggest insecurity comes from there not being guidance on how to document your approach, because everything, of course, in clinical trials needs to be documented quite well. But if you find ways of really writing down a good rationale for it, if you can document this well for yourself and you are confident that you can defend your rationale as well, then I think you are set up for success here. And of course, in terms of us Europeans, there's always a limitation in terms of data protection with regards to remote source data review or remote source data verification. And that's one I'm rather pessimistic on, that it would change in the near future. But I'm happy to let myself be surprised here. And it would be good to have more access to data also remotely, and just be able to implement good processes to do it. As an outlook, for sure, we will need, and I'm sure we will also get, more guidance and more insurance also from regulators that this approach is well. There's just a lot of demand for this. And I think regulators themselves just struggle to some extent still with trying to find a good means of defining exactly what they would like to see. And getting good guidance on how to document it, how to approach it, I think this is something that would be very beneficial. And I think this would give a lot of reassurance to the people for actually implementing these approaches. As sponsors, well, we've already seen that they embrace risk-based monitoring more. I am quite sure this trend will continue, although as restrictions have become less and less frequent too, we have seen also, again, an uptick of desire to perform source data verification and trials in which maybe this is not entirely necessary, but I think this is also a process that will just take some time. CROs, I think, are also, of course, heavily impacted. They should be driving this push for risk-based monitoring, and they need to support both the sponsors with it, but also the study sites and their own employees with it. It is a period in which a lot of things change, and it is something that cannot just be left to its own devices. There needs to be a good plan strategy. So to wrap this up, we have once more the risk-based monitoring definition. So we have an adjustment of monitoring frequency and intensity based on risk factors we define. We start with the risks, so identifying risks in the processes, identifying risks in the data that is collected, identifying risks in the assessments that are performed, and then identifying also data sources to gain oversights over those risks. Then, based on these risk profiles, you implement risk-based monitoring by finding a good rationale to combine the different monitoring techniques. that we talked about today. And of course, very important, you should adapt during the clinical trial. Risk-based monitoring lives from continuous adaptations, from reviewing the risks that you have, from adjusting the monitoring intensity and frequency if you see high-performing sites or low-performing sites, and really setting yourself up for this flexibility as well.
Okay, and that's it of part 3 of our web seminar on risk-based monitoring. We hope now you have a good overview of what risk-based monitoring is and how it can benefit your clinical trials. If you have any further questions, comment down below or reach out to us at GCP-Service. Also, feel free to subscribe to not miss out on our future videos, and please feel free to join our free and ongoing web seminar series in which we feature many different topics in clinical research.